Hello everybody, welcome back to the third and final part of Lecture 9 of the Experimental Design and Analysis section of Core Skills. And before the break, we're looking at two-way or two-factor analysis of variance, which gave rise to this slippery idea of the statistical interaction. So now I want to look a little bit more at that same concept in another example, help you get your head around it, and essentially to reach this learning outcome, which is to be able to recognize and explain statistical interactions. And then we'll finally go on to briefly touch on problems with independence in analysis of variance type designs. So often in an experiment, the statistical interaction is the thing that you're interested in, much more than the main effects of the different factors or covariates, as they're called, so continuous predictors that you have in a particular analysis. So I want to tell you a bit of an amusing story about an experiment in which I was aiming to look specifically for a statistical interaction. In fact, actually, the example I gave you before the break is really also like this, in the sense that the main purpose of that experiment was to look for an interaction between exercise and age. So in this experiment, I was rather lucky to do my fieldwork in Sydney. So I did a postdoc in Sydney when I was younger, and I arrived in Sydney and my boss out there said, "Oh great, we've got a brilliant field site for you to do your field work." I was working on caterpillars, and he said, "Yeah, this particular beach is the best place to go because there's loads of these caterpillars in the trees behind the beach." And at the time I didn't realise that this was Sydney's #1 gay nudist beach. So I mean, that's brilliant. Sure, it's great fun on the beach. I was obviously in the woods at the back of the beach, not really paying attention to the beach itself, but I was wandering around on my own looking for caterpillars in the trees, and that did, it turned out, mean that I received quite a lot of attention from gentlemen who might be interested in people wandering around in the woods on this particular beach. Anyway, it was great fun, and they were all very polite when they discovered that I was just looking for caterpillars. The experiment was about the social lives of caterpillars, actually. So it's very common in nature to see caterpillars hanging around in big groups like this, and this is the species that I studied, called the black slug cup moth, rather ridiculous name. They're really horrible. They actually sting. These yellow spines produce a kind of nettle sting type effect, and when you, like I was, clambering around in the trees with them, they tend to fall down your shirt and they sting you and they're horrible. But I was interested in why they hang around in groups, because instinctively it's a sort of silly thing to do as a caterpillar, because you make yourself very conspicuous, and so surely therefore you make yourself vulnerable to predation. But the idea we had for this particular species, and this might be representative of many species of caterpillar, is that they hang around in groups in order to actually defend themselves against predation, that they produce some kind of coordinated defense behavior. And in fact, actually, when you attack these guys, what they all do is they rear up on their hind legs, as it were, and wiggle themselves around, and they wave these noxious spines towards the predator. And so as a group, they're quite a formidable thing to attack if you're a small bird, for example, which might be the sort of thing that normally attacks these things. So maybe actually working as a group, you might be more effective defending yourselves than you would be on your own. So that was basically the idea that I was interested in exploring in this experiment. Do caterpillars hang around in groups in order to give themselves more protection against predation? I should say the other main obvious cost of hanging around in a group is, as you can see on this slide, they're competing for the same food. They're eating the same leaves. These are leaves of a tree, a bit like a eucalyptus tree. And you'd think hanging around in a big group means you're going to run out of food, but whatever the advantage of being in the group is, it must be good enough to outweigh that disadvantage. So we did an experiment in these little wooded areas at the back of the beach where we put caterpillars in known group sizes and exposed them to predation, or protected them from predation. So the way to protect them from predation was to put them in a cage that was fully kind of enclosed to stop any birds or indeed large insects coming in and attacking them. And then we manipulated group size by putting out groups of small groups, medium groups and large groups of caterpillars. And in this experiment, just like the previous one, there are 3 null hypotheses because we've got 2 factors and the interaction to worry about. So the first null hypothesis was that there would be no effective group size. So the thing that we're measuring here, by the way, is development rate. So rather than actually looking at survival, we're looking at how quickly the caterpillars grow. If you're able to just concentrate on feeding, you grow quickly. If you're having to spend a lot of your time worrying about predators, then maybe you're going to develop slowly because you're not going to eat very much food. And that was sort of an underlying idea that we knew about with these caterpillars already. So the first null hypothesis, that there would be no effect of group size on the development rate of these caterpillars. The second null hypothesis is that putting a cage around these caterpillars, protecting them from predators, would have no effect on their development rate. And the third null hypothesis, the one that we were actually really interested in, was whether the group size, was that the group size would have no effect on whether the caterpillars benefited from the cage. So you can express this in two different ways. You can say either the effective group size will not itself be affected by adding a cage, or you can say the effect of adding a cage will not itself be affected by what group size you were dealing with. So what we're getting at here is, if being in a big group is all about protecting yourself from predators, then we would expect that if you're inside a cage and there are no predators, you're not going to see any benefit of being in a big group. Whereas if you're outside the cage, if you're not protected from predators by an artificial cage, then only the big groups maybe would be the ones that would be able to develop quickly, because the small groups would be too busy spending all their time fighting off predators. So here are the data, which we actually published from this study, and the result was really quite neat. And what you can see here is that it's not trivial in the sense that it's complicated, because you've got lots of different groups going on, just like in the exercise example. So we've got 3 different group sizes: small, medium and large. I can't remember the exact sizes, but it might be like 50 caterpillars, 20 caterpillars and 5 or something like that. Then we've got blue versus green means predators or not. So green means there's a cage protecting them from predators. Blue means they're exposed to predation. And then our measure of development is body length. So they all start off at kind of the same size, and then we measure their body length later on. And if they've grown a lot, then that suggests that they've fed a lot and that they've not been worrying about predators too much. So what do we see? We see for the large groups of caterpillars very little difference between those growing up inside a cage and those not. But for the small groups of caterpillars, they've grown almost twice as long if they were in the cage with, if they were in the cage protected from predators. So it looks like the effect of group size depends on whether there's a cage present, or put it another way, the effect of the cage depends on what group size you're looking at. No effective cage here in large groups, big effective cage in small groups. That is a statistical interaction, and it's what we were looking for. But obviously we need to run an analysis of variance to check that this is really statistically true, as it were. So the first thing we find from the ANOVA is the p-value for the effective group size. And that's not significant, it's greater than 0.05. There's a 28% chance of observing this by chance. What is this? This is the main effective group size. So forget the colours of the bars, just ask yourself, overall, did the caterpillars grow more in the small groups, the medium groups, or the large groups? And the answer is, yeah, it's about the same, really. If you look at the sort of midpoint of each of these pairs, they're all pretty similar. So overall, no significant effective group size. So at this point, you might stop the study and say, oh, there's nothing interesting going on here, because actually group size doesn't really help the caterpillars. I mean, being in a big group is no good. But of course, there's more to it than that. So what about predators versus non-predators, no predators? So this is like cage versus no cage. Compare the blues and the greens, ignoring the group size. So just try and imagine the sort of average of all the blues and the average of all the greens. And what you can see is the greens are all higher than the blues. So on average, caterpillars grew more rapidly, they got to a longer body size if they were protected from predators, if they were inside a cage. And this is that effect of being able to feed most of the time instead of having to constantly fight off predators. Probably in this study, the key predators are actually wasps, which come in and try and attack caterpillars. So if you're a caterpillar and you have to kind of keep wriggling around and try and scare off this wasp, then you know you're in trouble and you don't spend as much time feeding. But the really interesting bit is the interaction. There's a significant interaction here, which means that the effect of the caging depended on the group size, as we can see rather nicely from the data. That for the small group sizes, we can see that being protected from the predators really helped, meant you could grow much more fast. or faster, as we say, whereas in the large groups, it had very little effect. And this is because of this thing about having to fight off predators. In the large groups, what happens is you're in a big gang all together, a predatory wasp arrives, a few of the caterpillars fight him off, and everybody else can carry on chewing on the leaf. Whereas if you're all dotted around individually, when your leaf gets attacked, you're the one that's got to fight off the predator or you're going to be in trouble. So the conclusion was that caterpillars hang around in large groups because it provides them with protection from predation. So that was the conclusion from the study, and it was all predicated on this key interaction that we were looking for. Okay, so that's analysis of variance and the interactions that arise when you have more than one factor. But the great thing about ANOVA is it can cope with much more complicated designs than that. You can have as many factors as you like, in fact. As soon as you add more than one factor, you've got the potential for interactions. If we add a third factor, then you've got interactions possible between all of the factors. So you rapidly get to a situation where there's quite a lot of stuff going on. So analysis of variance can get really rather complicated, difficult to interpret, but in theory you can chuck in as many factors as you have. So the last topic I want to look at is to go back to thinking about the assumptions of analysis of variance, which is a parametric statistical test that makes quite a lot of assumptions, but the really critical sort of fundamental assumption it makes is that the data are independent. And I talked about this at some length before when we were talking about t-tests, for example. And you can have a situation in a t-test where the data come in pairs, and so each data point in one group is paired with another data point in the other group. So the data in the two groups are not independent, they're related in some way. And that kind of non-independence can arise for all kinds of different reasons. And it can arise just as likely in ANOVA as it can in a t-test. So I want to go back to this example of the school kids. If you remember, we looked at an example where we were asking, does exercise improve learning in school kids? Because this is an oft said sort of not quite urban myth, but anyway, it's one of those things that people assume is true. But nobody had done the research actually until people in our School of Life Sciences in Neuroscience in Nottingham actually looked at this. And I described to you an experiment or a set of observations. Well, it was an experiment where kids were given exercise regimes to do, and then we looked at whether they were better at learning as a result of their test scores. Did their test scores go up? But I described to you an experiment in which there was a big problem, and that problem I called pseudo-replication. So I described to you an experiment where you have like a whole school full of kids, and you say, right, you're all going to do some mindfulness exercises, and you're not going to do physical exercise. And then you get another school and you say, right, you lot are all going to do some high energy exercise regime, and then we're going to test how well you've been learning in your classes. And if we find that this school is doing better than that school, we would think, oh, we can conclude that the exercise was really helpful. But the problem is that this study commits the sin of pseudo-replication, because each individual child in this top school shares in common not just the fact that they were doing the mindfulness regime, but also the fact that they went to that school. And if that school had a less effective teacher, or the facilities weren't as good, or the classes got interrupted by a fire alarm, then their learning could be worse than the other school by chance. In other words, by something which happened in that particular school. So the sample size, instead of being lots and lots of kids, you know, n equals 100 or something, the sample size is really only one, because you've only got one school in each case. And each school could be influenced by a whole variety of different factors. So pseudo-replication is when it looks like you've got replication, but you haven't. Or it looks like you've got lots of replication, but you've only got a bit. So I talked about, well, how do we solve this problem? And there are some obvious solutions. But a solution to this problem doesn't necessarily have to involve getting rid of all of these pseudo-replicates. So in this case, the pseudo-replicates are the kids, because there are lots of kids in each school, and they all share something in common, that school. So we could simplify things by just picking one kid at random within the school. But having lots of kids in the school is still useful. It's giving us more data in total, and it's giving us a better estimate of what's going on in that school. So just because it's not true replication which is relevant to the problem that we're tackling, it doesn't mean that it doesn't tell us something useful. So it's quite common to have experimental designs which look kind of like this, where you've got in, let's imagine, 3 schools and 100 kids in each of them that are being given the exercise regime, and 3 schools doing the mindfulness. So here the sample size is 3, really, 3 schools, not 300 kids. But we're still going to use all 300 kids, because having 100 kids in each school gives us a really good estimate of what's going on in each of those 3 replicates. And so it's really useful information. So we kind of get a hybrid here with some true replication, n equals 3, and then some pseudo replication, all those extra kids in each school. And this is an example of what's called a nested experimental design, where the pseudo replicates, the kids in our case, are kind of nested within the true replicates, which in this case are the schools. So imagine each yellow box is a school and each white blob is a kid. We've got lots of kids within each school, and then we've got multiple schools in each of the 2 treatments, the yellow treatment and the blue treatment, exercise and mindfulness. Or it could be, you know, this might be actually an aerial photograph of a study site where you've got fields with different crop plants growing in them, and the fields are either being treated with a pesticide or not. The 5 data points, whatever they are, in each of these blocks shares something in common, because they're nested within the true replicate, and they all share that particular individual replicate's identity, whether it be a school or a field or whatever. So this kind of design, like I say, it's quite common, and the nice thing is that there is a way of analysing what we've got here, looking for an effect of the yellows versus the blues, the training exercise, the exercise versus the mindfulness, at the same time as taking into account all of that pseudo replication. And the analysis you would use is called nested analysis of variance, or nested ANOVA, and it's really a subtle variation on exactly the kind of analysis I've already shown you in the last couple of lectures. I'm not going to say any more about it now, except that you need to be aware that different designs are possible, and really you need to understand how the replication in your study works in order to find the right kind of analysis when it comes to it. There are also one other sort of variation on ANOVA, which is very like this, that I'm going to mention, and it's called repeated measures ANOVA. And this is for situations that are like the kind of paired designs we looked at before. So where your data points are in some way related between the different treatments. And the most obvious way that they can be related is through time. So the kind of classic paired experimental design is a medical before and after trial. So you measure somebody's blood pressure and give them a drug and then measure it again. So you have a baseline and then a treatment measure, but it's from the same patient. So your data points in the control, if you like, or the before, are related to the data points in the after data set. And analysis of variance allows you to cope with variations on that, where you have more than 2 data points. So you might have a medical trial where you take regular measurements, like it might be a COVID vaccine trial where you're looking at antibody levels in patients every 3 weeks all the way through the year. So you have a whole series of data points that share something in common. They all come from the same patient, but they also come from different time points. So you can look for the effect of the time, taking into account the fact that the data points are related through the fact that they belong to the same patient. And that kind of analysis, you can't do it in a straightforward ANOVA design, a straightforward analysis of variance like we've used previously, because the data are not independent. But you can use a repeated measures analysis of variance to do the job for you. Again, not going to teach you the details of how it works, just be aware that such things exist, and they require you, or you are required, to think about independence before your study starts, really, in order to understand where the analysis might be heading. Okay, so that's the end of this week's lecture. Next week, it's the final kind of content lecture of this part of the course. There will be a revision session afterwards, but I want to deal with the last topic, which is regression, which is a form of analysis for trends rather than differences. We spent a lot of time talking about differences. I want to go back to thinking about trends. This is a trend that I have never been able to understand, the fashion for Ugg boots. I've been doing these lectures for many years. When Ugg boots were at the height of their fashion, which is like, I don't know, 10 to 15 years ago, everybody was wearing them in my lectures. I put this picture in my slides and I said I'm not going to remove it until my whole class no longer owns a pair of Ugg boots. And this year, unfortunately, I don't have a lecture theatre to ask, but every year until now, people have had Ugg boots, so I'm sticking with it. It's still a trend I don't understand. In Australia, where I have spent a bit of my time, my wife's Australian, Ugg boots, which come from Australia originally, are like the height of dagginess, as they would say. You know, like it's so uncool to wear Ugg boots, and yet they are really fashionable in Britain, or were anyway. Maybe not so much now. That's not really very relevant, except to say that the lecture is going to be about trends. So we'll look at regression and some related concepts next week.